morphological disparities like legs coming out of the head of a fly, the antennapedia mutation. These aren't promising avenues for future evolutionary development. So, just saying the words evo-devo doesn't solve the problem. If that's part of your evolutionary theory, that's not adequate from our point of view either. I am an advocate of intelligent design. We've debated on that topic before, and we stayed on that topic. I'm happy to come back to do that. I see evidence of intelligent design in the origin of life. I've written a book about that. I don't think it's an argument from ignorance. I think it's an argument from positive evidence, from the evidence of the information and the information processing system that's in living systems. That's an artifact of one and only one type of cause based on our experience, and that causes intelligence. I'm perfectly prepared also to say that the best explanation for the Cambrian explosion, what I call the Cambrian information explosion, also involves intelligence. I don't think it's an inherently disreputable idea to think that an intelligent agent, even a deity, infused information into the history of life at some point. That's only disreputable if you presuppose that there can't be such an entity. If you see evidence of the distinctive activity of intelligence in the history of life, why not talk about it? Why not infer it? So I don't think beginning to think about whether there's other instances of design or other occasions in which design might have been suffused into the environment, I don't think that's an inherently disreputable thing. And your line of questioning suggests that we should at this point fold up because we might have to admit that the design occurred more than once. Well, so what? Why is that disreputable? If there's a designer, that entity could have acted on more than one occasion.
Okay, let me rebut that very quickly here. First, to the point that was argued about the flukes in the whale. We actually have data on that. Okay, the fossil record is actually surprisingly good on that. It turns out because you look at Ambulocetus and many of the early whale fossils, they're like most mammals. They actually move in an up and down motion of their spine when they run. And Ambulocetus has vertebrae which show clearly that it has an up and down motion not only in its spine but all the way back to the end of its tail. There's no possibility, given the canalization that these animals have, that they're ever going to have a side-to-side motion of the tail for swimming. The fluke is already constrained to be that way. And also, in answer to what Dr. Sternberg said, one of the things he doesn't mention about that sequence of fossil whales is that every one of those fossils has a slightly more advanced condition in many features. They don't all do it all at once. Some of the early features appear as early as the terrestrial pachycetids, which have already got aquatic ear regions and all sorts of other features, and yet they're pretty terrestrial looking in their body skeleton. Others appear in later fossils. It's a stepwise mosaic process. It's not all within 2 or 3 lineages in that particular thing. So it's not like, and if we had the soft tissues, we probably could see more there, but that's where evo-devo comes in. It allows us to see how these soft tissues might have come in packages and how they're controlled by very similar mechanisms. So it's not inexplicable, especially if you use modern thinking rather than obsolete ideas about population genetics and extrapolism. The second question, again, is if Dr. Meyer keeps on adding his intelligent design argument there, he's retreated out of science once again. It's supernatural. It's not a testable hypothesis because he will not allow it to be falsified.
It is testable, and it's testable in the same way all historical scientific theories are testable. You ought to know about the method of historical scientific reasoning. I show in the book, Signature in the Cell, that the case for intelligent design is based on the same method of scientific reasoning that was used by Darwin and by Lyell. And that method is a method called the method of multiple competing hypotheses, standard in geology, where I was also trained. And it is and the hypothesis of intelligent design is testable by comparing its explanatory power over and against its competing hypotheses, and it's also testable by comparing the predictive success of the theory. The theory, for example, has predicted that the so-called non-coding regions of the genome, which were said to be junk by the Darwinists when it was first discovered, and was affirmed to be junk by Michael Shermer as recently in his 2008 book. Intelligent design advocates have been predicting since the early 90s that the non-coding regions of the genome would be shown to be importantly functional. Those predictions have been confirmed. In fact, overall, the non-coding regions of the genome...
Excuse me, that's incorrect.
That is correct.
Incorrect.
No,
I have it right in front of me here.
I don't think you want to argue about junk DNA with Richard Sternberg at my side. He's writing a book about it.
I'm happy to.
Yeah.
Okay. I did a whole bunch of research on this.
We don't have time for that, I'm afraid. I'm going to have to go
to questions.
Anyway, intelligent design has made a prediction about junk DNA not being junk that makes it scientific. We think the evidence has come in overwhelmingly. In fact, it's not we. It's coming in the scientific literature every week that shows that the junk is not junk. It's importantly functional.
Okay, let me answer that one before it gets passed over. It turns out, yes, the early naive Darwinists before this discovery of nucleotide sequencing thought everything was functional, and so they were shocked to discover in the 1960s with the very beginning of DNA sequencing and actually from the very primitive techniques they had, like gel chromatography, that there was a lot more DNA than seemed to be needed. And the basic thing is that there's a whole series of things in the DNA that clearly cannot be functional, that are junk. We don't know how much is junk, but enough of it is that it's clearly non-functional. I'll bring up one that was thrown at Dr. Meyer in Oklahoma a few weeks ago and apparently didn't know how to answer, and that's endogenous retroviruses. These are retroviruses that insert their DNA back into your cells when they infect you or when they infected your distant ancestor. They lock that in, and then your cells carry this. It's sort of like a scar tissue. You carry this dead virus DNA generation after generation after generation, no longer capable of producing those viruses, is evidence that your distant ancestors were once infected and it no longer has a function. And everybody who works on that agrees there's no possible functionality to this. It's completely non-coding. Pseudogenes are also once function genes that have lost their coding. Maybe some portion of what we used to think was non-functional turns out to be functional, but most of it apparently is non-functional and non-coding. You can see that very clearly if you look at things like how much DNA it takes to make an organism. Ferns and salamanders, which are, most of us concede, a little bit simpler than us, have 100 times more DNA than we do. There's even an amoeba, which is called Protopolychaeus debium, which has 200 times the DNA of a human, just to make an amoeba, okay? Clearly not much of that is in use to make something as simple as an amoeba. An onion, one species of onion has 5 times the DNA that humans have, another species has 20 times. These are two species in the same genus Allium. Clearly, that can't all be functional DNA if you have that much variation within a genus and that much variation from the simplest amoeba all the way to things like frogs and salamanders. So there's a lot of DNA that's not coded for, and I would say that majority, by far, of the people who actually work on this would say that most DNA is still non-coding.
Dr. Prothero, you're a geologist. We have a genomics expert to my right who'd like to comment.
I also had a degree in biology, and I've done a lot of work on this.
This is a competition between who's got more of a degree. You'll have the final comment, and we're going to go to questions.
Okay, I want to respond on the retroviral sequences topic. He's absolutely right. There are hundreds of thousands of copies of these virus-like sequences in our chromosomes of mammals and fishes and even the yeast cell. Now, what's interesting is that he's wrong on a number of points. Here's the first point. One of the first class of sequences to be expressed shortly after the first or two-cell embryo, that is from the zygote, including our zygotes, mouse zygote, to the two-cell stage embryo, first class of sequences are the endogenous retroviruses. And you know, as determined in 2004, is that if you knock those sequences out, you block their expression, you stop embryogenesis at the 4-cell stage. Actually, it's cited in a paper that Jim Shapiro and I, one of the papers published in 2005, where we dealt with this topic, this whole issue of why this so-called junk DNA is functional. Here's another example. What holds the placenta together is a class of proteins, and they form tethers. And the proteins that are coded are specified by retroviral-like sequences. It's different in the mouse than the ones in the human. If you look now, there are these switches that flip gene sequences on and off. They're called promoters. You can think of them as molecular toggle switches. A recent analysis of the number of these genetic switches in our genome contributed by these endogenous retroviruses. So many of them, they just have to be garbage. 181,000, roughly. So, and you look at the literature, what they're doing now, and this is, the ENCODE project was published in 2007, a series of papers where they looked at not just the sequences, but the expression of sequences, how they're copied in RNA. And what they found, to their surprise, is that most of the expression is taking place in this so-called non-coding DNA. And you can find papers in Nature and Science, genome research, genome biology. Thousands of them are out there now. Go to PubMed.org again, just type in junk DNA and see what you pull up. Don't take my word for it. But what you'll find is that when they investigate these classes of sequences, as numerous as they are, they're finding a whole series of functions. Pseudogenes, for example, so-called pseudogenes,